In a shallow embayment on the western edge of Australia, an invisible force once determined how much carbon the ocean could lock away, how firmly coastlines resisted erosion, and how entire underwater prairies grew or withered. That force was not a storm, not a temperature shift, not a human machine. It was the presence of a predator moving through clear water. The tiger shark altered ecosystems not merely through the bodies it consumed, but through the fear it projected. In regions such as Shark Bay, the simple possibility of encountering this hunter reshaped seagrass meadows across thousands of square kilometers. Carbon storage rose or fell depending on whether its shadow passed overhead. When that shadow fades, the consequences ripple outward from turtle grazing scars to atmospheric chemistry. This is not a story about predation alone. It is about intimidation as an ecological engine. The tiger shark is built for authority in shallow seas. Its body is broad and muscular, tapering to a powerful caudal fin capable of sudden acceleration. The head is blunt, housing wide jaws lined with serrated teeth shaped like curved blades, each capable of slicing through bone, shell, and cartilage. Unlike more specialized predators, this species is anatomically versatile. It consumes fish, rays, turtles, seabirds, and marine mammals. The digestive system tolerates hard carapaces and dense collagen. The sensory array is equally refined. Electroreceptors embedded along the snout detect faint muscle contractions from buried prey. The lateral line registers subtle pressure waves in turbid water. Vision functions effectively in the bright shallows, where seagrass thrives. This combination of strength and sensory acuity allows the shark to patrol wide territories that overlap with feeding grounds of large herbivores such as dugongs and green turtles. The crucial mechanism, however, is not constant attack. It is unpredictability. Tiger sharks do not need to kill every day in a given meadow to exert control. Their hunting pattern is mobile and opportunistic. They traverse channels, edges, and open flats in irregular intervals. From the perspective of a grazing turtle, this irregularity creates uncertainty. The turtle cannot predict whether the next stretch of exposed seagrass is safe. Dugongs, which are large marine mammals specialized for cropping seagrass rhizomes, face similar risk in open areas. Because these grazers must surface to breathe, they are constrained in where they feed. When sharks frequent shallow, open meadows, herbivores shift toward sheltered patches near mangroves or complex terrain that provide escape cover. The biological mechanism is behavioral modification under predation risk, a phenomenon known in ecology as a risk-mediated trophic interaction. Energy intake declines slightly. Grazing becomes patchy. Entire feeding landscapes reorganize around perceived danger. The first escalation emerges at the level of plant physiology. Seagrasses are flowering plants that evolved to live submerged in saline water. They anchor themselves through extensive rhizome networks that stabilize sediment. When heavily grazed, especially by dugongs that excavate below-ground tissues, seagrass beds can lose structural continuity. Recovery depends on time, nutrient availability, and light penetration. In Shark Bay, researchers observed that in areas where tiger sharks were abundant, turtles and dugongs avoided long exposed feeding bouts in central flats. These zones experienced lower grazing intensity. Shoots grew taller. Root mats thickened. Organic carbon accumulated in sediments beneath the meadow. In contrast, during periods when shark numbers declined, grazers returned to open areas and fed more intensively. The physical act of uprooting and repeated cropping reduced biomass and destabilized sediment layers that had stored carbon for decades. The second escalation unfolds in the chemistry of the seabed. Seagrass meadows are among the most efficient carbon sinks in the marine environment. Through photosynthesis, they fix carbon dioxide dissolved in seawater and incorporate it into tissues. A substantial fraction of that carbon becomes buried in anoxic sediments beneath the root zone, where decomposition proceeds slowly. When grazing pressure intensifies and rhizome mats fragment, oxygen penetrates deeper into sediments. Microbial respiration accelerates. Previously stored carbon can be remineralized and released back into the water column as dissolved carbon dioxide. In shallow coastal systems, this carbon may eventually exchange with the atmosphere. The presence of a predator thus modulates not only plant distribution, but also sediment biogeochemistry. Fear influences how much carbon remains locked beneath the seabed. A third escalation involves hydrodynamics and coastal protection. Dense seagrass meadows dampen wave energy. Their blades reduce current velocity, allowing suspended particles to settle. The root network binds sediments, decreasing erosion. When overgrazing thins these meadows, sediment becomes more mobile. Turbidity increases, reducing light penetration and further stressing remaining plants. The feedback loop can shift a stable meadow into a degraded state dominated by bare sand. Shorelines adjacent to these systems become more vulnerable to wave action. In regions where coastal communities rely on natural buffers to protect infrastructure, the decline of a predator can indirectly heighten erosion risk. The tiger shark's influence extends from the behavior of herbivores to the physical resilience of coasts. Another layer of consequence appears in biodiversity patterns. Seagrass beds provide nursery habitat for juvenile fish and invertebrates. Structural complexity offers refuge from predators and supports epiphytic algae and invertebrate communities. When grazing is spatially heterogeneous, as it is under high shark presence, mosaics of tall and short seagrass create diverse microhabitats. This heterogeneity supports a wider range of species with different shelter and feeding requirements. When grazing becomes uniformly intense, habitat complexity declines. Recruitment of commercially important fish species may fall. Local fisheries feel the effect not through shark bites, but through altered plant architecture. The connection to human civilization becomes clearer when these ecological processes are viewed at scale. Coastal carbon sequestration contributes to global climate regulation. Although seagrass occupies a relatively small fraction of the ocean floor, its capacity to bury carbon per unit area exceeds that of many terrestrial forests. In a warming world, maintaining and restoring blue carbon habitats has become a strategy for mitigating atmospheric carbon dioxide concentrations. The regulation of these habitats by apex predators adds an additional layer of complexity. Shark declines driven by overfishing and bycatch can cascade into reduced carbon storage potential. The economic value of this lost carbon sequestration is difficult to quantify precisely, but it intersects with global carbon markets and climate policy discussions. Fisheries are also entangled in this web. In some regions, the removal of large sharks leads to increases in mesopredator populations, such as rays. These mesopredators may consume shellfish at higher rates, affecting commercial harvests. In seagrass systems, altered grazing patterns can influence the abundance of species targeted by local fishers. Tourism industries that depend on clear waters and vibrant marine life may suffer if meadows degrade and turbidity rises. The tiger shark, often perceived primarily as a threat to swimmers, becomes a regulator of ecosystem services upon which coastal economies depend. A surprising fact deepens the narrative. Tiger sharks are not permanent residents in all shallow bays. Many individuals undertake seasonal migrations spanning hundreds of kilometers. Their movement patterns respond to temperature gradients, prey distribution, and reproductive cycles. This mobility means that the landscape of fear is dynamic. In seasons when sharks are abundant, grazers adjust behavior accordingly. When sharks migrate elsewhere, herbivores expand into previously risky zones. The system oscillates between states of high and low predation risk. Such temporal variation can enhance resilience by preventing any single grazing pattern from dominating indefinitely. The predator's migratory behavior thus introduces pulses of ecological regulation. The twist, however, lies in the vulnerability of the regulator itself. Tiger sharks mature slowly and produce relatively few offspring compared to many bony fish species. They are frequently caught as bycatch in longline and gillnet fisheries. In some regions, they are targeted for fins and meat. Because their ecological impact relies partly on their mere presence, even moderate declines can weaken the landscape of fear effect. Herbivores, sensitive to risk but also to energetic needs, may recalibrate their behavior if encounters become rare. Over time, Grazing intensity in open meadows can increase enough to tip the balance toward degradation. A predator that once stabilized carbon-rich habitats through intimidation becomes scarce, and the system drifts toward a different equilibrium. Another unexpected layer emerges from climate change itself. Rising sea temperatures can alter shark distribution and prey availability. Heat stress events can directly damage seagrass through physiological thresholds related to photosynthetic enzymes and membrane stability. If warming reduces seagrass resilience, while shark populations are simultaneously pressured by fishing, the combined stresses could amplify meadow collapse. The predator that once indirectly buffered carbon storage might no longer exert sufficient control in a changing ocean. The anomaly is not simply that a shark influences climate-related processes. It is that this influence depends on a delicate balance of movement patterns, reproductive biology, and human management. Research in Shark Bay demonstrated that areas with sustained shark presence maintained higher seagrass biomass and carbon stocks compared to zones experiencing reduced predator pressure. The mechanism was not continuous predation, but chronic risk perception. This distinction challenges simplistic views of food webs as linear chains of consumption. Instead, behavior-mediated interactions can be as powerful as direct feeding links. The tiger shark shapes space through anticipation. Dugongs and turtles calculate exposure risk. Seagrass responds to altered grazing. Sediments store or release carbon accordingly. Each step is biologically grounded, yet the aggregate effect touches planetary cycles. The final image is not of teeth, but of water moving gently over a meadow. Sunlight filters down, illuminating blades swaying in a current slowed by their own density. Beneath them, layers of dark sediment hold carbon accumulated over generations of growth. Somewhere beyond the edge of visibility, a tiger shark glides through a channel. Its presence is enough. Turtles hesitate before venturing into open flats. Dugongs alter their feeding paths. The meadow breathes, unravels carbon from seawater, and buries it in silence. When that shadow disappears from the system, the hesitation fades. Grazing scars widen. Sediment loosens. Carbon begins to move again. The ocean does not simply contain predators. It deploys them as agents that restructure landscapes through both action and anticipation. The tiger shark stands as an anomaly in the truest sense. A creature feared for its bite exerts planetary influence through restraint. By shaping the behavior of herbivores, it modulates plant growth, sediment chemistry, coastal stability, and carbon flux. Its shadow stretches from shallow bays to the atmosphere itself. In the quiet oscillation between fear and feeding, ecosystems are written and rewritten. A crustacean the length of a human hand forced engineers to rethink how to build machines that survive impact. In laboratories far from coral reefs, researchers redesigned body armor, aircraft panels, and protective sports equipment because of a strike delivered in a fraction of a blink beneath tropical water. The Mantis shrimp does not simply break shells. It altered the direction of materials science by demonstrating that strength alone is insufficient. Structure matters more than mass. Architecture defeats brute force. A reef predator evolved to shatter armored prey, exposed weaknesses in how humans once designed protection, and in doing so, reshaped technologies that guard soldiers, athletes, and spacecraft. Inside shallow coral reefs, where limestone frameworks rise from clear water, the mantis shrimp occupies burrows carved into rock or sediment. It is not large, but its forelimbs are transformed into raptorial appendages, unlike any other in the animal kingdom. In so-called smashers, the terminal segment forms a dense calcified club. This club is accelerated by a latch and spring mechanism embedded within the limb. Muscles contract slowly, storing elastic energy in specialized saddle-shaped structures composed of mineralized chitin. A latch holds the appendage in place as energy accumulates. When released, the limb rotates forward with extraordinary acceleration, striking prey with a velocity comparable to high-caliber projectiles. The impact generates forces sufficient to fracture snail shells and crab carapaces that would otherwise resist compression. The mechanism does not rely solely on muscle power. It exploits elastic recoil. This is critical. Muscle tissue has limits to contraction speed. By loading energy into a biological spring, the mantis shrimp surpasses those limits. The rapid strike also creates cavitation bubbles in the surrounding water. As the club moves forward, pressure drops behind it, forming vapor-filled cavities that collapse violently upon contact with the target. The collapse produces additional shock waves and brief flashes of light known as sonoluminescence. In effect, prey experience a double assault. Direct mechanical impact, followed by implosive forces generated by the water itself. The biological system integrates muscle, mineral, and fluid dynamics into a single destructive event. What astonished engineers was not merely the power of the strike, but the resilience of the weapon. Repeated impacts at such intensity should fracture the club itself. Yet mantis shrimp strike thousands of times across their lifespan without catastrophic failure. Microscopic analysis revealed why. The club is composed of multiple layers arranged in a helicoidal pattern. Chitin fibers are embedded in a mineral matrix of calcium phosphate. Each successive layer is rotated slightly relative to the one beneath it, creating a spiral stacking known as a boulogane structure. When cracks initiate under stress, they are deflected and twisted along curved paths, rather than propagating straight through the material. Energy is dissipated gradually, preventing sudden breakage. The outermost surface is densely mineralized for hardness, while underlying layers are more compliant, absorbing shock. It is a graded composite engineered by evolution. The first cascade effect unfolded not in the reef, but in research institutions studying impact resistance. Traditional armor design often emphasized homogeneous hardness. Steel plates, ceramic inserts, and rigid composites were optimized to resist penetration. However, homogeneous materials can fail abruptly once cracks form. The Mantis Shrimp Club demonstrated that layering and controlled crack deflection can dramatically increase toughness without excessive weight. Engineers began to experiment with fiber-reinforced composites arranged in rotated microlayers, mimicking the helicoidal architecture. The insight shifted thinking from simply resisting force to managing it. Materials could be designed to channel energy along predetermined paths, reducing catastrophic fracture. A second escalation occurred in aerospace engineering. Aircraft and spacecraft components must withstand repeated stress cycles, debris impacts, and temperature fluctuations while remaining lightweight. Inspired by the mantis shrimp, researchers explored composite panels with graded stiffness, where outer surfaces resist abrasion and inner layers absorb shock. The concept of hierarchical structuring, observed in the crustaceans club, influenced the development of laminates that combine high-strength fibers with resin matrices arranged in rotated sequences. By studying how biological materials integrate mineral and organic components, engineers refined approaches to fatigue resistance. The reef predator's anatomy thus entered wind tunnels and orbital design discussions. Another surprising dimension of the mantis shrimp lies in its visual system, which complements its striking ability. Although the focus here is impact mechanics, the animal's eyes possess specialized photoreceptors capable of detecting polarized light and a broad spectrum of wavelengths. This advanced visual processing aids in prey detection and communication. For materials scientists, the mantis shrimp became a symbol of integrated design, where sensory systems, mechanical structures, and behavior co-evolve to optimize performance. The realization that natural systems solve multiple engineering problems simultaneously influenced interdisciplinary research. Biologists, physicists, and engineers collaborated more closely, using the mantis shrimp as a case study in bio-inspired innovation. The ecological cascade within the reef is no less intricate. By preying on hard-shelled organisms such as snails and crabs, mantis shrimp regulate populations that graze on algae or scavenge detritus. When shell-crushing predators are abundant, certain gastropod populations remain controlled, preventing overgrazing on coral surfaces. This balance contributes to reef resilience. If mantis shrimp numbers decline, shifts in invertebrate communities may alter competitive dynamics between algae and corals. While they are not apex predators, their specialized feeding behavior influences trophic interactions at a localized scale. The same strike that inspired protective gear also maintains micro-ecological equilibrium. human civilization intersects with this organism in unexpected ways. Protective equipment for military personnel has incorporated lessons from the Mantis shrimp's club architecture. Ceramic armor plates combined with fiber backings are engineered to spread impact energy, reducing blunt force trauma. Sports helmets increasingly rely on multi-layer composites designed to manage rotational forces and dissipate shock, rather than merely resist penetration. In aerospace, lightweight panels inspired by helicoidal layering reduce structural failure risks while maintaining fuel efficiency. These innovations affect safety standards, manufacturing costs, and performance metrics across industries. A crustacean living among coral rubble influences procurement decisions in defense departments and design choices in commercial aviation. The economic implications extend further. Research into bio-inspired materials stimulates new patents, manufacturing techniques, and industrial partnerships. Universities secure funding to study natural composites, leading to collaborations between marine biology departments and engineering firms. The Mantis shrimp's anatomy becomes a template for additive manufacturing processes that print layered structures with controlled fiber orientation. This cross-pollination between ecology and industry demonstrates that biodiversity functions as a library of design solutions. When species are lost, potential innovations may vanish before being understood. Thus, the value of reef organisms cannot be measured solely by fisheries yield or tourism revenue. A new tension emerges when considering the vulnerability of coral reef habitats. Climate change drives ocean warming and acidification, stressing corals and altering community composition. Reef degradation reduces habitat complexity, potentially affecting mantis shrimp burrow availability and prey distribution. If reef structures collapse, populations of these crustaceans may decline. The species that inspired advances in impact-resistant materials depends on fragile carbonate frameworks threatened by rising temperatures and changing chemistry. The irony is profound. Human technologies, some developed to mitigate or survive environmental challenges, draw inspiration from organisms endangered by those same global pressures. Another unexpected layer involves the limits of biomimicry. While engineers replicate aspects of the Mantis shrimp's club, scaling biological architectures to industrial production presents challenges. Synthetic composites must endure conditions far beyond those encountered in shallow reefs, including extreme temperatures and prolonged mechanical loads. The translation from biological microstructure to industrial macrostructure requires adaptation. Nonetheless, the core principle remains transformative. Instead of designing materials as uniform blocks, engineers increasingly view them as complex assemblies of layers, gradients, and orientations. The crustacean's evolutionary experiment becomes a blueprint for innovation, even as its natural environment faces uncertainty. There is also a philosophical shift embedded in this story. For decades, human engineering often pursued maximal rigidity and strength. The mantis shrimp demonstrates that controlled flexibility and crack management can be superior strategies. This insight alters design thinking beyond armor and aerospace. It influences architecture, automotive safety, and even biomedical implants where shock absorption and fracture resistance are critical. The organism's weapon reframes the question from how to stop force to how to distribute and dissipate it intelligently. In this sense, the reef crustacean reshaped not only materials but mindset. The final image returns to the reef. In a narrow burrow within coral rubble, a mantis shrimp waits, its compound eyes scanning the water column. A snail inches across the substrate, unaware of the stored energy coiled within the predator's limb. Muscles contract slowly. Elastic structures load with potential. The latch releases. The club arcs forward, water cavitates, shell fractures. The prey becomes sustenance. Far above, in laboratories and factories, layered composites are tested against ballistic impacts and mechanical stress. Panels flex, cracks twist, energy disperses along spiraled pathways modeled after a crustacean's appendage. The connection between reef and runway, between burrow and body armor, is not symbolic. It is structural. The ocean does not merely host curiosities. It engineers solutions through millions of years of selective pressure. The mantis shrimp's punch reverberates beyond the reef, influencing how humans protect themselves and explore the skies. A small predator armed with a layered club demonstrates that innovation is not confined to workshops or research centers. It emerges from tidal flats, coral ledges, and evolutionary arms races. In studying the strike that shatters shells, humanity discovered new ways to withstand impact. The anomaly lies not in the violence of the blow, but in the architecture that survives it, and in the realization that the sea continuously designs technologies long before humans learn to recognize them. A population of dolphins in a remote corner of Western Australia forced science to redraw one of its oldest boundaries, the line between instinct and culture. For centuries, humans defined themselves by the ability to transmit knowledge socially across generations, to build traditions that outlive individuals. Yet in the shallow waters of Shark Bay, a group of bottlenose dolphins demonstrated that culture does not belong exclusively to humanity. They altered the way intelligence is defined, not through language we can hear, but through behavior that spreads across social networks without a single gene changing. The impact is philosophical, biological, and profound. An ocean predator reshaped how science understands the mind. In the clear, seagrass-rich shallows of Shark Bay, female bottlenose dolphins dive toward the seabed, carrying marine sponges fitted over their rostrums. These sponges are not prey. They are tools. The dolphins detach them from the substrate, position them over their snouts, and use them as protective coverings while foraging along the bottom. Beneath the sand lie small fish and invertebrates that lack swim bladders, making them nearly invisible to echolocation. To locate them, dolphins disturb the sediment with their rostrums. The sponge acts as a buffer against abrasion and sharp objects such as broken shells or stingray spines. This behavior requires a precise sequence of actions: selecting an appropriate sponge, detaching it without destroying its structure, transporting it and manipulating it while searching for hidden prey. It is not a reflex. It is a learned foraging strategy. The anatomical foundation of this behavior lies in the dolphin's sensory system. Bottlenose dolphins rely heavily on echolocation, emitting clicks and interpreting returning echoes to construct a three-dimensional acoustic map of their surroundings. However, certain prey species evade detection by lacking reflective gas-filled organs. These prey remain concealed within sediment layers. By using a sponge as protective armor, dolphins can probe deeper and more aggressively into the substrate. The sponge absorbs mechanical stress and reduces the risk of injury. The dolphin's elongated rostrum, densely innervated and sensitive, can otherwise suffer cuts or punctures. The integration of tool use with echolocation and tactile exploration demonstrates a coordinated cognitive and sensory process. The dolphin is not merely reacting to stimuli. It is executing a learned technique adapted to a specific ecological niche. What distinguishes this behavior is not only its complexity, but its transmission pathway. Genetic analyses reveal that sponging dolphins are not genetically distinct from non-spongers. The trait is not inherited through DNA. Instead, it spreads primarily through social learning, especially along maternal lines. Calves observe their mothers for years before becoming independent. During this extended juvenile period, they adopt the sponge-carrying technique by imitation. Not all dolphins in Shark Bay practice this method. Only certain lineages within specific social clusters do so. These clusters form what can be described as subcultures, defined by shared behavioral traditions rather than genetic separation. The ocean becomes a mosaic of knowledge communities. The first cascade effect emerges within the ecosystem itself. Sponging dolphins target prey that other dolphins rarely exploit. By accessing buried fish species, they reduce competition with non-sponging conspecifics. This niche specialization allows multiple foraging strategies to coexist within the same population. Resource partitioning reduces direct conflict and may stabilize local prey dynamics. In this way, culture influences ecological structure. A learned behavior reshapes feeding patterns, which in turn affect the distribution and abundance of benthic organisms. The ripple moves outward from cognition to community composition. A second escalation appears when examining social networks. Dolphins that practice sponging associate preferentially with one another. Shared behavior strengthens social bonds. Social structure becomes partially organized around cultural traits. In effect, knowledge determines companionship. This parallels patterns observed in certain primate societies, where tool use or vocal dialects define group identity. The presence of culture influences mating patterns, alliance formation, and cooperative behavior. Intelligence here is not an isolated attribute. It reorganizes social architecture. The implications for evolutionary biology are significant. Traditional models emphasize genetic mutation and natural selection as primary drivers of adaptation. Cultural transmission introduces a parallel pathway. Behaviors can spread rapidly without waiting for genetic change. If environmental conditions shift, dolphins capable of innovating and teaching new techniques may adapt more quickly than populations relying solely on inherited traits. This creates a feedback loop between cognition and survival. The capacity for learning becomes a selective advantage, potentially influencing brain size and neural complexity over generations. A surprising dimension of this discovery lies in its methodological impact on science itself. For decades, researchers hesitated to attribute culture to non-human animals, fearing anthropomorphism. The Shark Bay dolphins forced a reassessment. Long-term field studies combining behavioral observation, genetic sampling, and social network analysis provided robust evidence that the sponge-carrying technique spreads through learning rather than heredity. This challenged the assumption that culture requires language in a human sense. It expanded the definition to include socially transmitted, group-specific behaviors that persist over time. Marine mammals, once viewed primarily through the lens of instinct and reflex, entered the realm of cultural beings. The cascade extends further into the concept of intelligence. Intelligence is often measured through problem-solving tasks in controlled environments. Yet the dolphins of Shark Bay demonstrated intelligence embedded within ecological context. Their tool use is not performed for experimental reward. It enhances foraging efficiency in a specific habitat. This situational intelligence suggests that cognition evolves in response to environmental complexity. The shallow banks of Shark Bay, with their patchy seagrass and hidden prey, create selective pressure for innovative strategies. Intelligence becomes inseparable from landscape. Human civilization intersects with this story in multiple ways. Dolphins have long captured human imagination, influencing art, mythology, and tourism industries. However, the recognition of non-human culture carries deeper consequences. Conservation strategies must account not only for population numbers, but for cultural diversity within populations. If a subgroup practicing unique foraging traditions is lost, the knowledge may disappear permanently. Protecting biodiversity thus includes preserving behavioral repertoires. In regions where coastal development, boat traffic, or fishing activities disrupt dolphin habitats, culturally distinct groups may be disproportionately affected. The loss would not simply reduce headcounts, it would erase traditions. There are economic dimensions as well. Shark Bay is a World Heritage Site, partly because of its unique marine life. Dolphin tourism contributes to local economies. Scientific interest in dolphin cognition attracts research funding and international collaboration. The cultural behaviors of dolphins elevate their status beyond charismatic megafauna. They become subjects in debates about animal rights, ethical treatment, and marine protected area design. Policy discussions increasingly consider the cognitive capacities of marine mammals when regulating noise pollution, naval sonar, and fishing gear entanglement. The philosophical shift triggered by tool-using dolphins influences legislation and public perception. Another escalation arises when comparing dolphin cultures across regions. In other parts of the world, dolphins display different learned behaviors, such as cooperative strand feeding or unique vocal dialects. Each population carries distinct traditions shaped by local ecology. This suggests that culture in dolphins is not an isolated anomaly, but a broader phenomenon. The ocean contains multiple knowledge systems operating simultaneously. The diversity of these systems may enhance resilience at a species level. If one region faces environmental disruption, other populations with different strategies might persist. Cultural variation becomes a buffer against change. The twist in this narrative is that such cultural systems are fragile. Social learning requires stable maternal bonds and extended juvenile periods. Dolphins have relatively slow reproductive rates and long lifespans. Human pressures such as bycatch, habitat degradation, and pollution can remove key individuals from social networks. If experienced mothers are lost, calves may fail to acquire specialized techniques like sponge carrying. Cultural extinction can precede biological extinction. A population may survive numerically, while losing the knowledge that once defined its identity. The ocean's intelligence can erode silently. Climate change introduces additional uncertainty. Rising sea temperatures and shifting prey distributions may alter the availability of the buried fish species targeted by sponging dolphins. If ecological conditions change faster than cultural adaptation can respond, established traditions may become obsolete. Dolphins may innovate new strategies, but innovation carries risk. The very cognitive flexibility that once allowed them to redefine intelligence now becomes a test of survival under accelerating environmental change. In the end, the image returns to the shallow waters of Shark Bay. A mother dolphin descends with a sponge balanced on her rostrum, her calf trailing close behind. She presses the sponge into the sand, stirring sediment clouds that shimmer in filtered sunlight. A hidden fish darts upward, seized in an instant. The calf watches, memorizing movements that will shape its future. Above them, boats pass at a distance, and beyond the horizon, scientific debates unfold about culture, consciousness, and the uniqueness of the human mind. The lesson remains submerged yet unmistakable. The ocean does not merely house intelligence. It cultivates it, transmits it, and weaves it into ecosystems. Dolphins altered scientific thought not through conquest or dominance, but through shared knowledge carried across generations. In their submerged classrooms, without written language or constructed tools beyond a sponge, they demonstrated that culture is not confined to land. It flows through seawater, carried in memory and imitation. The anomaly is not that dolphins use tools. It is that they forced humanity to confront a new reality. The boundary we once believed separated us from other minds was never as solid as we imagined. In tropical coastlines across the Indo-Pacific, a single step in shallow water has altered emergency medicine protocols, reshaped public health messaging, and driven the development of one of the most specialized marine antivenoms in existence. The stonefish does not hunt humans, yet its venom has rewritten how coastal societies prepare for injury. Hospitals in reef-lined regions stock specific antivenom because of this animal. Lifeguards are trained to recognize its sting pattern. Tourism boards issue warnings that reference its shape. Military medical manuals include it in tropical deployment briefings. A camouflaged reef predator, almost invisible against coral rubble, forced modern medicine to adapt to a toxin delivered not in pursuit of prey, but in defense. The biological mechanism behind this impact begins with anatomy so deceptively passive that it borders on invisibility. Stonefish rest motionless on the seafloor, their bodies encrusted with algae and textured to resemble rock. Their dorsal surface bears a row of stout spines, each connected to venom glands embedded in thick tissue at the base. When pressure is applied from above, as when a foot descends unknowingly onto its back, the spines are depressed downward. This mechanical action compresses the venom glands, forcing toxin upward through grooves in the spines and into the puncturing wound. The system requires no conscious strike. It is a pressure-activated injection device evolved for instantaneous defense. The venom is a complex mixture of proteins, including potent cytotoxins and neurotoxins that disrupt cell membranes, interfere with ion channels, and trigger intense pain through rapid activation of nociceptors. Once injected, the venom acts with extraordinary speed. Victims report immediate, excruciating pain radiating from the wound site, often accompanied by swelling, tissue necrosis, and in severe cases systemic symptoms such as hypertension, cardiac arrhythmias, and muscle paralysis. The molecular mechanism involves pore-forming proteins that compromise cell integrity, leading to cell death and inflammatory cascades. The pain response is not incidental. It is biologically strategic. By inducing rapid incapacitation, the stonefish ensures that large predators learn to avoid stepping or biting. The venom is defensive, but its potency far exceeds what would be necessary to deter a single threat. It is an evolutionary overinvestment in survival. The first cascade effect unfolds along coastlines where humans and reefs intersect. In tropical regions dependent on tourism, shallow lagoons and coral flats attract swimmers and snorkelers. Increased human presence in these habitats raises the probability of accidental encounters. Each severe sting becomes not only a medical emergency but a public incident. Reports of stonefish injuries spread quickly, influencing perceptions of safety. Local authorities respond by installing warning signage, distributing educational materials, and training beach staff in first response protocols. The presence of this fish reshapes how coastal spaces are managed. Risk communication becomes integrated into tourism infrastructure. The escalation continues within medical research. Early accounts of stonefish envenomation revealed variability in symptom severity, prompting scientific investigation into venom composition and dose-response relationships. Researchers isolated specific protein components responsible for cardiovascular and neuromuscular effects. Antivenom development required immunizing animals with controlled venom doses to produce antibodies capable of neutralizing toxins. This process advanced understanding of marine venoms more broadly. Techniques refined during stonefish antivenom production informed research into other venomous marine organisms, from box jellyfish to cone snails. A single fish expanded the scope of toxinology. Another surprising layer of consequence appears in military medicine. Naval personnel and amphibious units operating in tropical waters face environmental hazards beyond conventional combat injuries. Stonefish envenomation is included in training curricula for medics deployed to reef-rich regions. Field manuals describe symptoms, first aid measures such as hot water immersion to denature heat labile toxins, and criteria for antivenom administration. The stonefish thus influences preparedness planning in defense institutions. Its venom has entered the language of operational risk assessment. Ecologically, the stonefish occupies a role as an ambush predator feeding on small fish and crustaceans. Its camouflage allows it to remain undetected by prey until the final moment of suction feeding. By regulating populations of small reef fish, stonefish contribute to local trophic balance. Their presence may influence prey behavior, altering microhabitat use among reef organisms. While not apex predators, their defensive venom allows them to occupy exposed positions without constant predation pressure. This defensive advantage stabilizes their ecological niche. The venom that injures humans primarily functions to maintain their place in the reef food web. The cascade extends into public health policy. Regions with frequent stonefish encounters integrate venom awareness into broader coastal safety campaigns. Educational programs teach residents and tourists to shuffle feet when walking in shallow water to avoid stepping directly onto camouflaged fish. Emergency response systems in tropical hospitals include protocols for rapid assessment and antivenom access. The cost of maintaining antivenom stockpiles, training personnel, and disseminating information represents an ongoing investment driven by the biology of a single species. Health systems adapt not only to infectious diseases but to venomous fauna embedded within local ecosystems. A further escalation arises from climate change and shifting species distributions. As ocean temperatures warm, the range of certain reef species may expand into new areas. Reports of stonefish sightings outside historically documented zones prompt re-evaluation of coastal hazard maps. Communities previously unfamiliar with this fish may encounter it for the first time. Medical facilities in these regions must consider whether to stock antivenom and train staff. The movement of a venomous species across geographic boundaries transforms regional health planning. The ocean's thermal gradients indirectly shape emergency medicine logistics. The influence of stonefish venom also intersects with biomedical research beyond antivenom. Certain protein components exhibit properties that interest pharmacologists studying ion channel modulation and pain pathways. While the venom causes severe pain in its natural context, controlled investigation of its molecular structure provides insights into how pain signals are transmitted. Understanding these mechanisms contributes to broader efforts in pain management research. A toxin evolved for defense becomes a subject of inquiry in the quest to alleviate suffering. The twist in this narrative lies in the stonefish's own vulnerability. Despite its formidable venom, the species depends on intact reef habitats for camouflage and prey availability. Coral bleaching, driven by rising sea temperatures and ocean acidification, degrades reef structure. As coral cover declines, the complex topography that conceals stonefish diminishes. Increased sedimentation from coastal development further alters habitat quality. The fish that shaped coastal medicine may itself be at risk from environmental change. If reef systems collapse, stonefish populations could decline or shift distribution, altering the pattern of envenomation incidents. The very hazard that prompted medical innovation is tethered to fragile ecosystems under stress. Another unexpected dimension involves human harvesting. In some regions, stonefish are captured for consumption, their venomous spines carefully removed. This practice requires specialized knowledge and carries risk. Overfishing or targeted collection can affect local population densities. Changes in abundance influence both ecological interactions and injury statistics. A reduction in stonefish numbers may lower sting incidence, but could also alter reef prey dynamics. The relationship between human exploitation and venom risk is not straightforward. Management decisions must balance ecological considerations with public safety. The broader philosophical implication emerges when considering how a defensive adaptation radiates outward into social systems. The stonefish did not evolve to influence hospital supply chains or tourism policies. Its venom is a localized evolutionary solution to predation pressure. Yet in coastal societies, intertwined with reef ecosystems, that solution becomes a driver of institutional behavior. Emergency departments adjust protocols. Governments fund toxin research. Educational campaigns shape human movement patterns along beaches. The ocean's biological strategies intersect with governance and infrastructure. The final image settles again on a shallow reef flat at low tide. Sunlight filters through warm water, illuminating a surface that appears to be scattered rock. Among the rubble lies a stonefish, motionless, dorsal spines poised like a concealed barricade. A swimmer's foot hovers inches above, unaware of the biochemical arsenal below. In distant laboratories, vials of antivenom sit refrigerated, antibodies prepared for rapid deployment. Manuals on tropical medicine reference the fish by name. Warning signs along beaches depict its silhouette. The chain of cause and effect extends from that still body on the seabed to medical training rooms and policy meetings. The ocean does not passively host hazards. It generates them, refines them, and projects their influence into human systems. The stonefish's venom is not merely a biological weapon. It is a catalyst that reshaped coastal medicine and public health awareness. A creature designed for camouflage and defense became an architect of emergency response frameworks. In its silence beneath the reef, the stonefish demonstrates that the sea's adaptations do not remain confined to water. They surface in hospitals, in legislation, and in the collective memory of communities that live at the edge of coral and sand. The anomaly is not the sting itself. It is the way a hidden fish compelled entire societies to reorganize around its presence. Across tropical coastlines, a reptile once hunted nearly to extinction silently governs the stability of underwater carbon banks that influence climate regulation, shoreline protection, and fisheries productivity. The green sea turtle does not build reefs or produce oxygen directly, yet its grazing patterns determine whether vast seagrass meadows thrive or decay. When populations of this turtle collapsed under centuries of hunting, entire underwater landscapes shifted. Carbon storage declined. Sediments loosened. Shorelines became more vulnerable to erosion. The impact was not dramatic in a single season. It unfolded quietly, through the absence of teeth trimming blades of grass beneath the tide. A reptile shaped the architecture of coastal ecosystems, and by extension, the resilience of human settlements that depend on them. The biological mechanism begins with anatomy refined over millions of years for herbivory in saltwater environments. The green sea turtle possesses a serrated, beak-like jaw adapted not for tearing flesh, but for cropping vegetation. Its powerful neck muscles allow it to grasp seagrass blades and shear them cleanly near the base. Unlike many marine herbivores that browse intermittently, green sea turtles feed consistently, moving across shallow meadows and grazing patches in a manner similar to terrestrial ungulates. Their digestive systems host specialized microbial communities capable of fermenting plant material rich in cellulose. This symbiotic gut microbiota enables them to extract nutrients from fibrous leaves that would otherwise be indigestible. The turtle's foraging behavior is deliberate and methodical, focusing on younger, nutrient-dense shoots that grow near the sediment surface. Seagrass itself is not a simple marine plant. It is a flowering angiosperm adapted to life submerged in saline water. It forms dense meadows anchored by rhizomes that bind sediment and create complex root networks. When left ungrazed for extended periods, seagrass blades can grow long and accumulate epiphytic algae and sediment on their surfaces. Excessive growth reduces light penetration to lower leaves and can increase susceptibility to disease. By cropping the blades regularly, green sea turtles stimulate new growth. The act of grazing removes older tissue and promotes the production of fresh shoots with higher photosynthetic efficiency. In effect, the turtle prunes the meadow, maintaining it in a productive, early successional state. The first cascade effect emerges within the meadow structure. Regular grazing creates a mosaic of short, dense patches interspersed with slightly taller areas. This heterogeneity enhances habitat complexity. Juvenile fish and invertebrates find refuge within the varied canopy heights. Nutrient cycling accelerates as cropped plant material decomposes and releases nitrogen and phosphorus back into the sediment. The turtle's feeding patterns influence not only plant morphology, but also microbial activity in the root zone. Oxygen released by healthy seagrass roots into surrounding sediment supports aerobic bacteria that process organic matter efficiently. Without grazing, overgrown blades can collapse, shading lower layers and reducing root oxygenation. The meadow transitions from dynamic growth to stagnation. A second escalation unfolds in carbon dynamics. Seagrass meadows are among the most effective carbon sinks in coastal ecosystems. Through photosynthesis, they capture carbon dioxide dissolved in seawater and convert it into biomass. A significant portion of this carbon becomes buried in sediments beneath the meadow, where low oxygen conditions slow decomposition. When green sea turtles maintain seagrass in a vigorous growth phase, carbon fixation rates remain high. Root systems expand, trapping additional sediment and organic material. In contrast, when turtle populations decline and grazing pressure diminishes, meadows can become overgrown and unstable. Excess biomass may detach during storms, exporting carbon away from the meadow rather than sequestering it. Sediment disturbance increases, exposing buried carbon to oxidation and potential release back into the water column. Another surprising layer of consequence appears in sediment stability. The intertwined rhizomes of seagrass act as a natural mesh that holds fine particles in place. Healthy meadows dampen wave energy and reduce current velocity, allowing suspended sediments to settle. This stabilization protects shorelines from erosion. When turtle grazing ceased in many regions due to overharvesting, seagrass meadows in some areas declined in quality. Thick mats of aging leaves collapsed under their own weight or were uprooted by storms. Without consistent cropping to encourage root reinforcement, sediment became more mobile. Increased turbidity reduced light penetration, further stressing remaining plants. A feedback loop emerged in which meadow degradation accelerated coastal vulnerability. The cascade extends outward into adjacent ecosystems. Seagrass meadows often lie near coral reefs and mangroves, forming interconnected coastal networks. Many fish species use seagrass as nursery habitat before migrating to reefs as adults. If meadows degrade, juvenile survival rates decline, affecting reef fish populations. This reduction influences fisheries yields and alters predator-prey dynamics in offshore systems. The grazing of a reptile in shallow water thus reverberates through food webs extending beyond the meadow itself. The turtle's feeding behavior links benthic plants to pelagic fisheries. Human civilization intersects directly with this ecological process. Coastal communities rely on stable shorelines to protect infrastructure, homes, and agricultural land. Seagrass meadows reduce wave energy before it reaches beaches, acting as natural barriers. The carbon sequestration provided by healthy meadows contributes to climate mitigation strategies, often referred to as blue carbon initiatives. Fisheries supported by seagrass nurseries sustain livelihoods and regional food security. When green sea turtle populations were heavily hunted for meat, shells, and oil, these ecosystem services were indirectly compromised. The removal of a grazer disrupted the maintenance system of underwater vegetation that underpins multiple economic sectors. The scale of historical exploitation was immense. In some regions, green sea turtles were harvested in numbers that reduced populations to a fraction of their former abundance. The absence of consistent grazing altered meadow dynamics over decades. Scientists observed areas where seagrass grew excessively long and then experienced sudden die-offs, leaving bare patches vulnerable to erosion. Carbon burial rates declined in certain degraded meadows. Restoration efforts focused initially on protecting seagrass itself, but research increasingly recognized that plant health was intertwined with turtle presence. Conservation strategies began to incorporate both habitat protection and turtle population recovery. A further escalation arises from climate change. Rising sea levels and increasing storm intensity place additional stress on coastal systems. Healthy seagrass meadows, maintained by turtle grazing, can adapt by expanding landward or increasing root density, enhancing resilience against wave action. Degraded meadows lack this capacity. The turtle's role becomes more critical under changing climatic conditions. Its feeding behavior supports the structural integrity needed to buffer coastlines against more energetic seas. The reptile, slow-moving and ancient in lineage, influences the future of rapidly growing coastal cities. The twist in this narrative challenges assumptions about conservation success. In some regions where turtle populations are recovering due to protective legislation, grazing intensity has increased markedly. In isolated meadows with limited spatial extent, concentrated grazing can lead to localized overcropping. The relationship between turtle density and meadow health is not linear. It depends on meadow size, nutrient availability, and predator presence. Sharks that prey on turtles create a landscape of fear, distributing grazing pressure across broader areas. Where shark populations have declined, turtles may graze more intensively in specific zones. The stability of the system relies on interactions among multiple species. The grazer that maintains meadows can, under altered predator dynamics, contribute to imbalances. Another unexpected dimension involves nutrient runoff from agriculture and urban development. Excess nutrients entering coastal waters stimulate algal growth that can outcompete seagrass. In nutrient-rich environments, turtle grazing alone cannot prevent meadow decline. The interplay between terrestrial land use and marine herbivory determines outcomes. Protecting turtles without addressing watershed management yields incomplete solutions. The reptile's ecological function operates within a larger network of environmental variables. Despite these complexities, the central reality remains that green sea turtles act as biological lawnmowers in underwater fields that store carbon and stabilize coastlines. Their jaws clip blades, stimulating renewal. Their movements distribute grazing pressure, shaping plant communities. Their digestive systems recycle nutrients. The absence of their presence leaves meadows prone to stagnation or collapse. The presence of their grazing restores dynamism. The final image settles beneath clear tropical water, where sunlight flickers across a dense seagrass meadow. A green sea turtle moves steadily, lowering its head to crop a patch of tender shoots. Each bite removes older tissue and encourages fresh growth. Beneath the blades, roots grip sediment that holds centuries of accumulated carbon. Beyond the meadow, waves break against a shoreline protected by this living buffer. Fishing boats rest in calm waters sustained by juvenile fish raised among the grass. The chain of influence extends from the turtle's beak to the stability of coasts and the chemistry of the atmosphere. The ocean does not simply host vegetation. It assigns maintenance to creatures that move through it with ancient purpose. The green sea turtle, often seen as a symbol of marine conservation, functions as an engineer of carbon infrastructure beneath the tide. Its grazing writes patterns into the seabed that determine whether carbon remains buried or returns to circulation. In the quiet rhythm of feeding, a reptile sustains systems that shield shores and moderate climate. The anomaly lies not in its survival through deep time, but in the realization that its daily meal shapes the resilience of a changing planet. Entire coastlines have shifted from towering underwater forests to silent stone deserts because of an animal no larger than a clenched fist. Where kelp once rose in dense canopies that slowed currents, sheltered fish, and drew carbon from the sea, there are now fields of exposed rock scattered with spines. Fisheries have collapsed. shorelines have become more vulnerable to storm energy. Carbon capture has declined. The transformation did not begin with a hurricane or a chemical spill. It began with a surge in sea urchin numbers. A small grazer, freed from the pressure of its predators, rewrote the structure of entire marine ecosystems. The biological mechanism is deceptively simple. Sea urchins are echinoderms equipped with a rigid internal skeleton of calcite plates and a mouth positioned on their underside known as Aristotle's lantern. This structure contains 5 continuously growing teeth arranged in a pentaradial symmetry. Each tooth is self-sharpening, composed of crystalline calcite reinforced with organic matrices that increase fracture resistance. The lantern protrudes downward, scraping and grinding plant tissue from hard substrates. Urchins feed primarily on macroalgae such as kelp, but in high densities they will also consume encrusting coralline algae and even juvenile kelp recruits. Their tube feet allow them to cling tightly to rock surfaces, resisting wave action while they graze. Under balanced ecological conditions, sea urchin populations are regulated by predators such as sea otters, large fish, lobsters, and certain starfish species. Predation keeps urchin density below the threshold at which grazing pressure overwhelms kelp growth. Kelp forests themselves are highly productive systems. These large brown algae anchor to rocky substrates with holdfasts and extend fronds upward toward the surface, forming 3-dimensional habitats that support fish, invertebrates, and marine mammals. Kelp growth depends on sufficient light, nutrient availability, and substrate stability. When urchins are present in moderate numbers, they consume drift algae and trim edges of kelp beds without eliminating the forest. The cascade begins when predators decline. Historical fur trade decimated sea otter populations in many regions. Overfishing reduced numbers of large predatory fish. In some areas, disease affected lobster stocks. With fewer predators removing them, sea urchin populations expanded rapidly. Urchins reproduce through broadcast spawning, releasing gametes into the water column where fertilization occurs externally. High densities increase the probability of successful fertilization events. Larvae drift with currents before settling onto rocky substrates. In predator-depleted systems, juvenile survival rates increase. The result is an exponential rise in grazing pressure, concentrated on kelp holdfasts and young fronds. Once grazing intensity surpasses kelp growth rates, a tipping point is reached. Urchins begin to aggregate at the base of kelp stalks, consuming the tissue that anchors the algae to rock. Whole plants detach and drift away. Without canopy cover, light penetration increases to the sea floor, but instead of promoting regrowth, the persistent presence of urchins prevents new recruits from establishing. The reef transitions into what scientists call an urchin barren. These barrens are characterized by low structural complexity, sparse macroalgae, and dense urchin populations. The system stabilizes in this degraded state because urchins can survive on minimal food, scraping residual algae and biofilms from rock. A surprising fact intensifies this transformation. Urchins in barrens can enter a state of reduced metabolic activity, shrinking their gonads and surviving for extended periods with limited nutrition. This physiological resilience allows them to persist even when kelp is scarce, maintaining pressure on any new growth. The barren becomes self-reinforcing. Kelp spores settle but are consumed before they can mature. The once towering forest is replaced by a flat, spiny landscape. The ecological consequences escalate rapidly. Kelp forests serve as nurseries for numerous fish species, including those targeted by commercial fisheries. Juvenile fish rely on the vertical structure of kelp fronds for shelter from predators and for access to prey. When forests collapse into barrens, recruitment declines. Fish populations decrease. Local fisheries experience reduced yields. The economic impact spreads to coastal communities dependent on these resources. A grazer smaller than a dinner plate triggers changes that reach fishing fleets and market supply chains. Another escalation emerges in carbon dynamics. Kelp forests are significant contributors to coastal carbon sequestration. Through photosynthesis, they fix carbon dioxide into biomass, some of which is transported offshore and buried in deep sea sediments. The removal of kelp reduces this carbon drawdown. Although kelp does not bury carbon in the same manner as seagrass, Its high productivity influences local carbon cycling and can contribute to long-term sequestration through detrital export. In barren systems, primary productivity declines sharply. The shift alters the balance of carbon flux along coastlines already stressed by climate change. Coastal protection is also affected. Dense kelp beds attenuate wave energy, reducing the force that reaches shorelines. Their flexible fronds absorb and dissipate kinetic energy from storms. In regions where kelp forests have collapsed into barrens, increased wave exposure can accelerate erosion of coastal bluffs and beaches. Infrastructure near shore becomes more vulnerable. The physical transformation of underwater vegetation has tangible effects above the tide line. The chain reaction extends further into trophic interactions. In kelp forests, diverse assemblages of invertebrates, fish, and marine mammals coexist within layered habitats. In barrens, biodiversity declines. Certain species adapted to open rocky habitats may increase, but overall complexity diminishes. Reduced habitat diversity can make ecosystems less resilient to additional stressors such as warming events or pollution. The simplification of food webs leaves fewer pathways for energy flow. The system becomes brittle. Human civilization intersects directly with these changes. In some regions, efforts to restore kelp forests have involved manual removal of urchins or reintroduction of predators. Sea otter recovery in parts of the North Pacific has demonstrated how reinstating a keystone predator can reverse barren formation. As otter populations rebound, urchin densities decline, allowing kelp to regrow. This dynamic illustrates the economic value of predator conservation. Ecotourism centered around otters generates revenue, while kelp restoration supports fisheries and coastal protection. The absence or presence of a small grazer thus influences policy decisions, marine protected area design, and resource management strategies. There is another surprising dimension. In some regions, climate change has compounded the problem by increasing sea temperatures and reducing nutrient availability. Kelp species are sensitive to prolonged warming. Heat stress weakens their growth and reproductive capacity. When warming coincides with high urchin densities, the likelihood of forest recovery diminishes. The combined pressure of climate and grazing pushes ecosystems toward persistent barren states. The urchin becomes not only a consumer, but an amplifier of climate vulnerability. The twist in this narrative lies in the dual identity of sea urchins. In balanced ecosystems, they contribute to biodiversity by grazing on algae and preventing dominance by a single species. Their presence can enhance patchiness and promote coexistence among different algal forms. It is only when predator populations decline or environmental conditions shift that urchins transform into agents of desertification. The same biological mechanism that maintains diversity under control can dismantle it under imbalance. The anomaly is not inherent malevolence. It is the amplification of small changes into systemic collapse. Efforts to manage urchin barrens have revealed additional complexity. In some areas, commercial harvest of urchins for their roe has created economic incentives to reduce densities. However, overharvesting in balanced systems can also disrupt local dynamics. Management requires understanding thresholds, predator interactions, and environmental variability. The interplay between fisheries economics and ecological stability underscores how closely human livelihoods are tied to the grazing patterns of a spiny invertebrate. The final image rests on a rocky coastline where cold currents sweep past submerged outcrops. In one cove, kelp towers sway in green columns, fish darting among fronds, sunlight filtered through layered canopies. In another, only stone and spines remain. Urchins clustered across barren rock, their teeth scraping at the last traces of life. The difference between these worlds is measured in predator presence, grazing intensity, and environmental balance. The transformation from forest to wasteland does not announce itself with noise. It unfolds through the steady motion of calcite teeth against kelp holdfasts. The ocean does not merely support life. It reorganizes it through interactions that magnify across scales. A sea urchin, driven by instinct to feed, can reshape coastlines, economies, and carbon pathways when freed from its constraints. The barren is not a static void. It is a testament to imbalance, to the cascading effects of removing predators and altering climate. In the quiet scraping of Aristotle's lantern, entire ecosystems pivot. The anomaly is not the existence of the grazer. It is the speed and magnitude with which its unchecked abundance can convert vibrant underwater forests into deserts of stone. In several regions of the world's oceans, the disappearance of large predatory fish has not left an empty throne. It has cleared the way for an entirely different kind of ruler. Squid, once woven quietly into the fabric of marine food webs, have surged in abundance in ecosystems reshaped by industrial fishing and warming seas. Their rise has altered commercial fisheries, redirected the diets of marine predators, and shifted the balance of oceanic energy flow. Some researchers describe this transformation as the emergence of cephalopod-dominated waters, a subtle but profound reorganization of marine life. A soft-bodied invertebrate, built for speed and rapid reproduction, has become a beneficiary of human-driven imbalance. The biological mechanism behind this ascent lies in the squid's anatomy and life history. Squid belong to the class Cephalopoda, possessing elongated bodies, 8 arms, and 2 specialized tentacles equipped with suction cups for capturing prey. Their mantle encloses powerful muscles that expel water through a siphon, generating jet propulsion. This allows rapid bursts of movement, enabling them to evade predators and ambush prey with remarkable agility. Their nervous systems are highly developed relative to other invertebrates, supporting complex behaviors, rapid camouflage through chromatophore expansion, and sophisticated hunting strategies. Unlike many long-lived fish species, most squid have short lifespans measured in months to a few years, depending on species. They grow quickly, mature early, and produce large numbers of eggs in a single reproductive season. This reproductive strategy is central to their ecological success. Squid populations can respond rapidly to favorable conditions. When predation pressure decreases or prey availability increases, recruitment can surge within a single generation. In contrast, many large predatory fish such as tuna or groupers require several years to reach sexual maturity and produce fewer offspring per reproductive cycle. Industrial fishing has disproportionately removed these slow-growing, late-maturing fish from many ecosystems. As top predators decline, predation pressure on squid decreases. At the same time, the removal of competing fish species can increase the availability of small forage fish and zooplankton that squid consume. The result is a demographic advantage rooted in life history traits. The first cascade effect unfolds within food webs. Squid occupy a middle trophic level, preying on crustaceans and small fish, while serving as prey for marine mammals, seabirds, and remaining predatory fish. As squid abundance increases, their predation on small fish intensifies. This can suppress populations of forage fish that are themselves critical prey for other predators. The energy that once flowed through long-lived fish species begins to route through shorter-lived cephalopods. Because squid grow quickly and die young, biomass turnover accelerates. Nutrient cycling rates change. The ocean's rhythm shifts toward faster pulses of growth and decay. Another surprising consequence emerges in predator diets. Marine mammals such as sperm whales and certain dolphin species have long consumed squid, but in some regions their dependence has increased as fish stocks decline. Seabirds adjust foraging patterns to exploit abundant squid near the surface. Even human fisheries pivot. As traditional fish stocks collapse or become economically unviable, commercial fleets increasingly target squid species. Global squid landings have risen in parallel with declines in some finfish stocks. Markets adapt, cuisines evolve, and supply chains reorganize around a different dominant resource. The escalation continues when considering ecosystem stability. Squid thrive in disturbed systems partly because they tolerate a wider range of environmental conditions than many fish species. Some squid exhibit flexible spawning times and can adjust migratory behavior in response to temperature shifts. Their ability to exploit variable habitats makes them resilient in warming oceans. As climate change alters sea temperatures and stratification patterns, certain squid populations expand into new regions. This expansion can introduce novel predator-prey interactions, further reorganizing local food webs. A fast-living invertebrate becomes a pioneer in altered seas. Carbon dynamics are also influenced. Squid, through their rapid growth and short lifespans, contribute to the biological pump in distinctive ways. When they die, their bodies sink, transporting organic carbon to deeper waters. In regions with high squid biomass, this process can represent a significant flux of carbon from surface to depth. However, because squid populations fluctuate dramatically, the carbon export associated with them is variable. In contrast, stable fish-dominated systems may exhibit more consistent patterns. The shift towards cephalopod dominance introduces volatility into carbon cycling processes, potentially affecting how ecosystems respond to climate variability. Human civilization is deeply entangled in this transformation. Coastal economies that once depended on predictable fish harvests must adapt to more variable squid fisheries. Squid catches can be abundant one season and scarce the next, reflecting environmental conditions and recruitment success. This variability complicates management strategies and economic planning. In some regions, the rise of squid fisheries has provided short-term economic relief following fishery collapses. Processing plants retool. Export markets adjust. Yet reliance on a species with boom and bust dynamics introduces new risks. There is another surprising layer. Squid are voracious predators capable of cannibalism under high-density conditions. In boom years, intense intraspecific predation can regulate population size. This self-limiting mechanism, however, does not necessarily restore former fish-dominated structures. Instead, it produces oscillations in abundance. Such oscillations reverberate through predator populations that depend on squid as food. Seabird breeding success may correlate with squid availability. Marine mammal migration patterns adjust to track shifting cephalopod concentrations. The system becomes more dynamic, less anchored by long-lived species. The twist lies in the fact that squid are not invaders from another world. They have always been part of marine ecosystems. Their recent prominence reflects the removal of constraints rather than the introduction of novelty. Overfishing, habitat degradation, and climate change have selectively disadvantaged slower-growing species. Squid, with their rapid life cycles and adaptive behaviors, are pre-adapted to capitalize on disturbance. They are beneficiaries of imbalance, not its architects. The anomaly is not their existence, but their opportunity. Another unexpected dimension involves evolutionary implications. In ecosystems where squid dominate prey availability, selective pressures on predators may shift. Species capable of efficiently capturing and digesting cephalopods may gain advantage. Over long timescales, dietary specialization could influence morphology and behavior in higher trophic levels. The ocean's restructuring under human influence may thus guide evolutionary trajectories in subtle ways. The rise of squid is not merely a demographic event, but a potential driver of future adaptation. From a management perspective, the concept of cephalopod oceans challenges traditional fisheries models. Management frameworks often assume relatively stable population structures and predictable recruitment patterns. Squid defy these assumptions. Their population dynamics are tightly coupled to environmental variability, such as ocean temperature, current patterns, and productivity cycles. Effective management requires integrating climate forecasting with harvest regulation. The growing importance of squid fisheries underscores the need for adaptive governance in a rapidly changing ocean. At the societal level, culinary trends and global trade patterns reflect this ecological shift. Squid, once considered a regional delicacy in certain cultures, have become staples in international markets. Processing technologies have improved to handle large, sometimes unpredictable catches. As fish stocks struggle under continued pressure, squid provide an alternative protein source for millions of people. Yet the sustainability of this reliance depends on careful monitoring of population trends and environmental drivers. The final image unfolds in open water at dusk. A shoal of squid pulses through the dim light, bodies flashing as chromatophores expand and contract in rapid succession. Beneath them, smaller fish scatter. Above, a seabird dives. In the distance, fishing vessels adjust nets designed for soft-bodied prey, rather than scaled fish. The water column feels different, alive with swift, short-lived creatures that mature and reproduce before the year turns. The ocean's architecture has shifted from forests of finned giants to clouds of jet-propelled hunters. The ocean does not remain static when its largest inhabitants are removed. It reorganizes. It redistributes energy. It elevates species whose biology matches the new conditions. Squid did not conquer the sea through dominance alone. They rose because disturbance favored their design. Their boom signals a broader transformation, one driven by human hands, yet expressed through natural life histories. In the flicker of chromatophores and the surge of jet propulsion, a new balance asserts itself. The anomaly is not that squid flourish. It is that their success reveals how profoundly human activity has reshaped the pathways of life beneath the waves. In the high Arctic, the disappearance of floating sea ice has forced tens of thousands of walruses onto crowded beaches, triggering mortality events visible from satellites and transforming a marine mammal into one of the most powerful living signals of planetary warming. When the ice retreats beyond the continental shelf where they feed, walruses abandon their traditional offshore resting platforms and haul out in dense masses along remote coastlines. Calves are crushed in stampedes. Adults overheat under prolonged sun exposure. Images of these gatherings circulate across the world, reframing climate change from abstract temperature graphs into the physical compression of bodies on shrinking land. A species that evolved to live between ice and water has become a barometer for the stability of the Arctic system. The biological mechanism that ties walruses to sea ice begins with anatomy and foraging ecology. Walruses are large pinnipeds, equipped with thick blubber for insulation, sensitive vibrissae that detect prey in sediment, and elongated tusks used for hauling their heavy bodies onto ice. They feed primarily on benthic invertebrates, such as clams, that inhabit shallow continental shelf waters. To access these prey, walruses dive repeatedly to the seafloor, using their whiskers to locate buried organisms and powerful suction to extract soft tissue from shells. Between dives, they require rest. Sea ice provides stable platforms positioned directly above feeding grounds, allowing energy-efficient foraging cycles. By resting on ice rather than swimming long distances, walruses conserve energy needed for thermoregulation and reproduction. As Arctic sea ice declines in extent and thickness due to rising air and ocean temperatures, the spatial relationship between ice and prey is disrupted. In late summer, ice retreats northward into deeper waters beyond the continental shelf. There, the seafloor lies too deep for efficient benthic feeding. Walruses follow the retreating ice until foraging becomes energetically impractical. At that threshold, they turn toward land, congregating on coastal beaches adjacent to remaining shallow feeding areas. These terrestrial haulouts can involve tens of thousands of individuals. The density of animals increases dramatically compared to dispersed ice platforms. The biological need for rest collides with the physical constraint of limited shoreline space. The first cascade effect is behavioral and demographic. On sea ice, walruses are distributed across wide areas, reducing crowding and the risk of disturbance-induced panic. On land, tightly packed groups are more susceptible to sudden movement. If startled by predators, aircraft, or human presence, animals may rush toward the water. Calves, smaller and less mobile, can be trampled by adults weighing over a ton. Mortality rates during such events rise. Increased calf mortality reduces recruitment into the population. Over time, this demographic pressure can slow recovery or contribute to decline. A surprising escalation emerges in energetic costs. Hauling out on land often requires walruses to swim greater distances between feeding grounds and resting sites compared to ice-based foraging. Extended swimming increases energy expenditure. For lactating females, higher energetic demand can affect milk production and calf growth. The interplay between ice availability and reproductive success introduces a subtle but consequential feedback. Reduced body condition may lower survival rates during harsh winters. The decline of ice alters not only where walruses rest, but how efficiently they convert prey into offspring. The cascade continues into benthic ecosystems. When large numbers of walruses concentrate near limited coastal feeding areas, localized depletion of clams and other invertebrates can occur. Walruses are ecosystem engineers of the seafloor, turning over sediment and redistributing nutrients as they forage. Concentrated feeding pressure may intensify sediment disturbance in specific zones, potentially affecting invertebrate community composition. These changes reverberate through Arctic food webs, influencing species that rely on benthic organisms. The redistribution of walrus foraging effort reshapes nutrient cycling patterns across the continental shelf. Another layer of consequence involves indigenous communities in the Arctic that have relied on walruses for subsistence for generations. Changes in haul-out locations and migration patterns affect access and safety for hunters. Increased coastal aggregation may bring walruses closer to some communities while moving them farther from others. Variability in ice conditions complicates travel routes and traditional knowledge systems built on predictable seasonal patterns. The biological response of walruses to ice loss intersects directly with cultural continuity and food security in Arctic societies. The visual impact of mass haul-outs has extended far beyond the Arctic. High-resolution imagery showing dense clusters of walruses on narrow beaches, sometimes accompanied by reports of trampling deaths, has become emblematic of climate change in public discourse. These scenes translate abstract data about temperature anomalies and ice extent into tangible narratives. The walrus, with its tusks and massive form, functions as a charismatic focal point in climate communication campaigns. Environmental organizations and media outlets have used such imagery to illustrate the urgency of warming trends. A shift in resting behavior becomes a symbol embedded in global awareness. Policy discussions have also been influenced. As evidence accumulated that sea ice decline affects walrus survival and reproduction, conservation status assessments were revisited. Debates over listing the species under protective legislation intensified. International bodies monitoring Arctic ecosystems incorporated walrus data into broader climate impact reports. The presence of mass haul-outs on land became a metric in evaluating the ecological consequences of ice retreat. The species entered geopolitical conversations about shipping routes, resource extraction, and emission reductions. A marine mammal shaped policy narratives across nations. Another surprising dimension lies in the feedback between climate change and communication. The walrus's visible behavioral shift provided a compelling case study that bridged science and storytelling. Unlike some Arctic species that remain elusive, walruses aggregate in ways that are readily documented. Their reliance on ice is direct and observable. As sea ice declines, the correlation between environmental change and animal behavior becomes visually undeniable. This clarity amplifies their role in shaping public understanding. The species becomes both subject and messenger. The twist in this narrative is that walruses themselves are not universally declining in all regions. In some populations, numbers have recovered from historical overhunting during earlier centuries. Conservation measures reduced direct human exploitation. Yet recovery in numbers does not equate to security in habitat. A larger population combined with reduced ice availability can intensify crowding on land. The very success of protective measures intersects with climate-driven habitat loss to create new vulnerabilities. The anomaly lies in the coexistence of demographic recovery and ecological instability. Another unexpected factor involves increasing human activity in the Arctic as ice retreats. Shipping lanes expand. Resource exploration intensifies. Noise from vessels and aircraft can disturb walrus haul-outs, increasing the likelihood of panic-induced stampedes. Thus, ice loss not only forces walruses onto land, but also exposes them to greater anthropogenic disturbance. The convergence of habitat contraction and human presence compounds risk. Mortality events become more probable in a warming, industrializing Arctic. From a physiological perspective, walruses are adapted to cold environments with thick blubber and limited capacity for heat dissipation on land. Prolonged haul-outs during warm weather can lead to thermal stress. On ice, animals can slip into the water to regulate body temperature. On crowded beaches, access to water may be constrained by social hierarchy and physical barriers. Heat stress interacts with crowding to create additional mortality risk, particularly for juveniles. Climate change thus influences both the physical substrate of rest and the thermal environment in which rest occurs. The final image returns to an Arctic shoreline under a pale summer sun. A vast assembly of walruses stretches across gravel and sand, their bodies pressed together in a landscape once dominated by floating ice. Beyond the beach, open water extends to the horizon where ice once lingered through the season. Calves nestle among adults, vulnerable in the density. In distant capitals, policymakers debate emissions targets, while satellite images of this gathering circulate on screens. The connection between atmospheric carbon and compressed bodies is not metaphorical. It is mechanistic. The ocean does not passively absorb heat. It translates warming into altered currents, thinning ice, and shifting habitats. Walruses respond according to biological necessity, seeking rest where ice no longer exists. Their congregation on land is not adaptation in triumph, but adaptation under constraint. In their mass haulouts, the Arctic broadcasts a signal. The anomaly is not that a species adjusts to change. It is that the scale of adjustment reveals the magnitude of transformation underway. Beneath the Arctic sun, thousands of tusks and flippers mark the edge of a climate boundary that continues to move. In tropical oceans, the disappearance of a fist-sized orange fish has signaled the unraveling of entire reef systems, shifting conservation funding, tourism economies, and global emotional investment in coral ecosystems. When sea anemones bleach and die under rising temperatures, clownfish vanish with them. Their absence is not merely aesthetic. It reflects the collapse of a tightly bound mutualism that stabilizes reef microhabitats. The fall of a brightly colored reef fish has become shorthand for the vulnerability of coral ecosystems under climate stress. A species once confined to shallow lagoons now occupies the center of global conversations about ocean warming. The biological mechanism that binds clownfish to their fate begins with a partnership that appears improbable. Clownfish live among the tentacles of specific sea anemone species, organisms armed with venomous nematocysts capable of paralyzing prey. Most fish that brush against these tentacles are stung and consumed. Clownfish survive because their skin is coated in a specialized mucus layer that prevents nematocyst discharge. This mucus either lacks triggering compounds or mimics the chemical signature of the anemone itself. Through gradual acclimation, juvenile clownfish learn to inhabit the tentacles without provoking a lethal response. In return for shelter, clownfish defend the anemone from predators and parasites, remove debris, and enhance water circulation around its tissues. The mutualism extends into nutrient exchange. Clownfish excrete nitrogen-rich waste products directly into the tentacle canopy, fertilizing the anemone and its symbiotic zooxanthellae. These microscopic algae reside within anemone tissues and perform photosynthesis, supplying energy to the host. When clownfish swim among tentacles, their movement increases oxygen flow and may stimulate photosynthetic efficiency. The partnership is thus multi-layered, involving chemical tolerance, behavioral defense, and metabolic exchange. Clownfish do not simply inhabit anemones. They integrate into their physiological processes. The first cascade begins when sea temperatures rise beyond tolerance thresholds for the zooxanthellae within anemones. Prolonged thermal stress disrupts photosynthetic pathways, generating reactive oxygen species that damage host tissues. In response, anemones expel their symbiotic algae, leading to bleaching. Without the energy contribution of zooxanthellae, anemones weaken and may die if stressful conditions persist. As anemones decline, clownfish lose both shelter and breeding sites. Clownfish are highly site-attached, often spending their entire lives within a single host anemone. When that host dies, individuals are exposed to predation and may fail to secure new territory. A surprising escalation emerges in reproductive dynamics. Clownfish live in hierarchical groups within each anemone, structured around a dominant breeding pair and subordinate juveniles. They exhibit protandrous hermaphroditism, meaning individuals begin life as males and can change sex to female if social rank shifts. The death of an anemone eliminates the spatial anchor that supports this social structure. Without stable hosts, breeding pairs may fragment. Recruitment of juveniles declines. Population crashes can occur rapidly following anemone mortality events. What appears to be the loss of a plant-like organism cascades into the collapse of a fish population governed by intricate social rules. The reef-level consequences extend further. Sea anemones occupy microhabitats among corals and rocky substrates. Their presence contributes to habitat heterogeneity. Clownfish, defending their host from butterfly fish and other anemone predators, indirectly influence local species composition. When bleaching events remove anemones, the protective behavior of clownfish disappears. Increased predation on remaining invertebrates may alter community structure in subtle ways. The removal of this mutualistic unit reverberates through the web of small-scale reef interactions that collectively sustain larger biodiversity patterns. Another escalation connects bleaching to reef resilience. Anemones, like corals, depend on symbiotic algae for energy. Widespread bleaching events that affect corals often impact anemones simultaneously. The decline of both organisms signals systemic thermal stress across reef ecosystems. Because clownfish are highly visible and charismatic, their disappearance can function as an early warning indicator for broader reef degradation. Monitoring clownfish abundance offers a proxy for assessing anemone health and, by extension, local bleaching severity. A small fish becomes a biological sensor embedded within coral communities. The chain reaction extends into human economies. Coral reefs support fisheries, tourism, and coastal protection. When bleaching events intensify, reef structure degrades, reducing habitat complexity for commercially valuable fish species. Tourism dependent on vibrant reef imagery suffers. The cultural prominence of clownfish, amplified through media and storytelling, has drawn public attention to these declines. Images of empty anemones where clownfish once darted evoke a tangible sense of loss. Donations to coral conservation initiatives often spike following widely publicized bleaching events that feature iconic reef species. Emotional engagement translates into funding streams that support research, restoration, and policy advocacy. A surprising dimension of this story lies in how a fictional portrayal amplified real-world impact. Popular media introduced clownfish to global audiences, embedding them in cultural consciousness far beyond tropical coastlines. As a result, the species became a gateway for reef education. Aquariums reported increased public interest in coral ecosystems. Educational programs leveraged familiarity with clownfish to explain symbiosis, bleaching, and climate change. The species functioned as an ambassador, bridging scientific complexity and public empathy. A reef fish influenced environmental communication at planetary scale. The twist emerges when considering the pressures generated by this popularity. Increased demand for clownfish in the aquarium trade surged after media exposure. In some regions, unsustainable collection practices threatened local populations. Although captive breeding programs have since expanded, initial spikes in wild capture added stress to already vulnerable reef systems. The same cultural visibility that drove conservation awareness also introduced new exploitation pressures. The symbol of fragility became subject to market forces. Another unexpected factor involves ocean acidification. As atmospheric carbon dioxide dissolves into seawater, it reduces pH and alters carbonate chemistry. While clownfish themselves are not calcifiers, their host anemones and surrounding corals are affected by shifts in carbonate availability. Acidification can impair coral growth and structural integrity, compounding bleaching impacts. Behavioral studies suggest that elevated carbon dioxide levels may influence sensory processing in some reef fish, potentially affecting predator avoidance and homing behavior. The interplay between warming, acidification, and habitat degradation creates a multi-layered threat matrix for clownfish populations. The social structure of clownfish adds further complexity. Because breeding depends on the presence of a stable anemone and hierarchical organization, recovery after bleaching may lag even if anemones return. Juveniles must locate suitable hosts and integrate into existing social systems. If host density declines below a threshold, spatial gaps increase and reproductive opportunities diminish. The resilience of clownfish populations is thus intertwined with spatial patterns of anemone recovery and larval dispersal. Connectivity between reef patches becomes critical. From a policy perspective, clownfish have influenced reef protection measures. Marine protected areas designed to conserve coral habitats often reference flagship species to justify boundaries and funding. Clownfish, recognizable and emotionally resonant, serve this function effectively. International climate negotiations cite coral bleaching as evidence of urgent need for emission reductions. The visibility of species dependent on coral and anemone health strengthens arguments for mitigation and adaptation strategies. A fish small enough to fit in a hand has entered the lexicon of global climate diplomacy. The final image settles on a reef at twilight. An anemone, once vibrant and pulsing with life, stands pale and diminished, its tentacles translucent under fading light. The familiar flash of orange and white is absent. In nearby waters, healthy anemones still shelter their resident fish, movements synchronized in quiet partnership. The difference between presence and absence is a measure of thermal stress and ecological balance. Above the reef, distant cities continue to emit heat-trapping gases that alter ocean temperatures thousands of miles away. The ocean does not merely endure warming. It expresses it through bleaching tissues, shifting partnerships, and silent vacated habitats. Clownfish do not generate headlines through size or strength. They do so through dependence. Their fate is inseparable from that of their hosts, and by extension, from the chemical equilibrium of seawater itself. The anomaly lies in how a small reef fish became a global emblem, translating molecular stress within anemone cells into a narrative that mobilizes funding, shapes policy, and reframes climate change in human terms. Beneath the surface, the disappearance of a flash of orange marks more than aesthetic loss. It marks a signal, broadcast from coral shallows to a warming world. Long before smokestacks altered the chemistry of the atmosphere, a deep-diving predator was quietly amplifying the ocean's ability to absorb carbon. The sperm whale, hunting in the black depths thousands of meters below the surface, acted as a vertical conduit between the abyss and the sunlit layer where microscopic plants draw carbon dioxide from the air. By feeding at depth and defecating near the surface, these whales redistributed nutrients in a way that stimulated phytoplankton growth, increasing carbon uptake and oxygen production. When industrial whaling decimated global populations, that nutrient conveyor weakened. The removal of a single predator reverberated upward through ocean chemistry and into atmospheric balance. A mammal of the deep influenced the air that circulates around the planet. The biological mechanism begins in darkness. Sperm whales descend into the mesopelagic and bathypelagic zones, diving for nearly an hour at a time in search of squid and deep-water fish. Their massive heads house an organ filled with waxy spermaceti, once harvested for oil, but biologically tied to buoyancy control and echolocation. Using powerful clicks that travel through water and return as echoes, they detect prey in near-total darkness. Their bodies are engineered for these dives, with collapsible lungs that withstand pressure and muscles adapted to tolerate low oxygen conditions. After feeding at depth, they ascend to breathe and rest near the surface, often remaining in the upper water column between dives. The crucial detail lies not only in what they eat, but in where they excrete. Prey captured in deep waters contain nutrients such as iron that are scarce in surface layers of certain ocean regions. Iron is a limiting micronutrient for phytoplankton growth in vast stretches of the ocean, particularly in areas known as high nutrient low chlorophyll zones. These waters contain macronutrients such as nitrate and phosphate, but lack sufficient iron to trigger large phytoplankton blooms. When sperm whales defecate near the surface, they release fecal plumes rich in iron and other nutrients. Unlike sinking detritus, these plumes are buoyant and remain suspended in the photic zone, where sunlight penetrates and photosynthesis occurs. Phytoplankton respond rapidly to this iron input. These microscopic organisms perform photosynthesis, using sunlight to convert carbon dioxide dissolved in seawater into organic matter. In doing so, they form the base of marine food webs and produce a substantial portion of the oxygen in Earth's atmosphere. Increased iron availability can stimulate phytoplankton growth, enhancing primary productivity. As phytoplankton proliferate, they absorb more carbon dioxide from the water, which in turn draws additional carbon dioxide from the atmosphere through gas exchange. The whale's feeding cycle thus links deep ocean prey consumption to surface carbon fixation. The first cascade effect unfolds in the biological pump, the process by which carbon fixed by phytoplankton is transported to the deep ocean. When phytoplankton die or are consumed by zooplankton, some of the resulting organic material sinks. A fraction of this carbon is sequestered in deep waters or sediments for long periods. By enhancing phytoplankton productivity through ion fertilization, sperm whales indirectly increase the flux of carbon to depth. This is not a linear process. It depends on species composition, grazing pressure, and water column dynamics. Yet the principle remains that whale-mediated nutrient redistribution can amplify carbon sequestration. A surprising escalation emerges when considering scale. Before industrial whaling in the 19th and 20th centuries, sperm whale populations were significantly larger than today. Estimates suggest that hundreds of thousands of individuals roamed the oceans. Each whale, through repeated dive cycles and surface defecation events, contributed to nutrient recycling. Cumulatively, these contributions may have enhanced regional carbon uptake to a measurable degree. The removal of large proportions of these populations through whaling likely reduced this fertilization effect. A predator targeted for oil and meat inadvertently influenced atmospheric chemistry. The cascade continues through trophic dynamics. By preying on large squid and fish, sperm whales regulate deep-water food webs. Their predation shapes the abundance and distribution of mesopelagic species. Changes in prey populations can alter nutrient cycling at depth. The whale's vertical movement integrates these layers, transporting energy and nutrients across gradients that would otherwise remain more isolated. This vertical coupling stabilizes ecosystem processes. When whale numbers decline, the intensity of this coupling diminishes. Another unexpected dimension involves oxygen production. Phytoplankton contribute a substantial fraction of global oxygen through photosynthesis. While terrestrial plants are often credited with oxygen generation, marine primary producers play an equally critical role. By stimulating phytoplankton growth, sperm whales indirectly support oxygen production. The magnitude of this contribution relative to global totals remains an active area of research, but the conceptual linkage is clear. A deep-diving mammal influences surface photosynthesis, which influences atmospheric composition. Human civilization intersects with this process through both history and climate policy. The era of commercial whaling extracted enormous biomass from the oceans. Ships pursued sperm whales across every major ocean basin, driven by demand for spermaceti oil used in lighting and lubrication. The ecological consequences of this extraction were not understood at the time. Today, as societies grapple with climate change and seek methods to enhance carbon sequestration, the role of marine megafauna in nutrient cycling has gained attention. Some researchers argue that restoring whale populations could contribute modestly to natural climate mitigation through enhanced carbon capture. A surprising economic angle arises from this perspective. Whale watching tourism now generates significant revenue in many coastal regions. Protecting and restoring whale populations supports local economies while potentially enhancing ecosystem services. The value of a living whale extends beyond tourism to include its ecological role in carbon cycling. Discussions of natural capital increasingly incorporate the services provided by large marine animals. The sperm whale's contribution to nutrient redistribution becomes part of broader conversations about ecosystem valuation. The twist in this narrative lies in complexity. The fertilization effect of whale feces depends on oceanographic conditions. In some regions, iron from terrestrial dust or upwelling may dominate nutrient inputs. In others, whale-mediated redistribution may be more significant. Moreover, climate change alters stratification patterns and nutrient availability, potentially modifying how effectively iron stimulates phytoplankton. The restoration of whale populations alone cannot reverse global carbon emissions. The system is intricate, with feedback loops that resist simplistic solutions. The whale is a component of a network, not a singular fix. Another unexpected factor involves whale carcasses. When sperm whales die, their bodies sink to the seafloor in events known as whale falls. These carcasses deliver substantial organic carbon to deep ecosystems, supporting specialized communities for decades. In pre-industrial oceans with larger whale populations, whale falls were more common, representing another pathway for carbon transfer to depth. The decline in whale numbers reduced not only nutrient recycling at the surface, but also carbon deposition in the deep sea. The loss of whales altered both upward and downward fluxes in the ocean carbon cycle. From an evolutionary perspective, the relationship between whales and phytoplankton illustrates co-evolutionary feedback across vast spatial scales. Phytoplankton support the food webs that ultimately sustain whales. Whales enhance phytoplankton growth through nutrient redistribution. This reciprocal interaction stabilizes productivity. The removal of whales disrupts that feedback, potentially reducing system resilience. In a warming ocean, resilience becomes increasingly critical. The final image unfolds in the open ocean under a high sun. A sperm whale surfaces, exhaling a column of mist that dissipates into the air. After several breaths, it arches and descends, flukes slicing the surface before vanishing into blue. Hours later, it returns, releasing a diffuse plume that drifts in the sunlit layer. Invisible to the naked eye, iron disperses among microscopic cells. Phytoplankton divide, drawing carbon dioxide from seawater, releasing oxygen into currents that circulate around the globe. The cycle continues, dive after dive, plume after plume. The ocean does not merely absorb carbon passively, it orchestrates exchanges through creatures that traverse its depths. The sperm whale is not a factory or a machine. It is a living link between abyss and atmosphere. Its feeding, diving, and defecation connect layers of the sea in ways that influence planetary chemistry. The anomaly is not that a whale eats squid in darkness. It is that this act, repeated across oceans and generations, once amplified the planet's capacity to breathe.